It's obviously music to my ears because anything to do with mRNA is an area where we're looking towards manufacturing the input proteins that you need to create mRNA. So any expansion of that market, whether it's in CAR T, cell and gene therapy, those are key areas where Dyadic can play and will play in the future, like our upcoming launch of DNase I.
Yeah. I thought I'd bring up something about Dyadic specifically because you guys have a couple of expression platforms, protein expression platforms. You've got C1, which has been around for quite a while, and then Dapibus, which is a little bit more recent. What's the difference between the two? I mean, aren't they just expression systems? Why do you need two of them?
That's a great point. Well, you need two of them because every time you engineer a cell line, you do some damage to it, which lessens its ability to produce protein. So, and that goes for any other expression systems as well. Anytime you do genetic engineering to a cell line, you are essentially hindering its capability to produce higher amounts of protein. The reason you do that is so you can produce more complex proteins. So C1 has been re-engineered from the industrial powerhouse that it was in the early 90s and 2000s to become a more highly evolved expression system to produce complex proteins. Where our Dapibus platform has been specifically designed and not engineered to the point where it can produce some of these complex proteins, but it retains a much higher yield potential than C1 does. So we're using C1 for the more complex and high value input protein markets, and we're using Dapibus for the lower margin, but also higher volume segments like food and nutrition and industrial.
Okay. Yeah, right, because I've seen that as well, the food nutrition side for Dapibus. I think you're working on some cheeses there, right?
Non-animal dairy proteins and enzymes, you're absolutely right, and they go into making cheese, milk products.
So, I think viewers are probably interested in this technology, just in general. What besides C1 is out there that produces proteins? Because viewers and investors realize that these are all produced somewhere, probably out in the ether. They don't really understand it, but there are a few choices that you can make to decide what you want to do to produce your monoclonal antibody or vaccine or other proteins like that. What are we looking at in terms of that spectrum?
I mean, it's a great question, and you're exactly right. There are many expression platforms that do many different things well, but they all have to produce correctly folded, modified, and functional proteins. So it doesn't matter what system you're using, that's the bar. If you can do it in higher yields and lower costs, you actually have an expression system that has better applicability in broader segments. That's obviously what we're trying to target. So if you look at things like monoclonals and vaccines or lipid nanoparticles, those are things that need to be made in either mammalian cell lines or E. coli cell lines or RC1 cell line. So you need a more evolved expression platform to produce those. Things like yeast, pichia, as well as trichoderma, other fungi, and our Dapibus platform are things that would be used for higher volume but lower margin segments like the industrial enzymes, food and beverage.
You mentioned some of the kind of the end markets there, and energy is actually another really interesting end market that you guys have with your partnership with Fermbox. What expression system are you using with them? Is that C1 or is that Dapibus?
Dapibus. Yeah.
Okay, because it's high volume. You don't need that precision that you would get with C1. Yes. So you want mass production. So tell us about what they're making. I mean, are they making ethanol type fuels or is it something else?
They're making the input proteins for the biomass processing that ultimately will make ethanol fuels.
Okay, so they're an earlier step in that.
Yes, the earlier step in the process.
Okay. Yeah, and I think that is also an interesting partnership because you might see some revenues, product-related revenues, from that. Tell us about what we might expect to see there.
It's a great point. It's one of the key tenets of our commercialization rollout, and the Fermbox did receive an order for a large amount of cellulosic enzymes in the first quarter of this year. They are currently producing and delivering as we speak. Should these enzymes be, they're going to be evaluated and used in application, and should they meet the application thresholds of the customer, there is a follow-on order for for a much larger follow-on order for the cellulosic enzymes. So that partnership is really starting to take flight. We're obviously excited about it, and the fact that it's direct product revenues, which is where we need to go as a company.
And that's going to be in Asia, I guess, where they're going to produce and sell those related products?
Yeah, currently in India, but yes.
Okay, in India. Okay.
Obviously be targeting broader areas.
Okay. Another interesting area is the mRNA and enzyme market. You've talked about that at Dyadic quite a bit as one of the kind of emerging areas that you want to pursue. How is that? How is that looking, and what are the next steps to move ahead on that side of things?
No, it's a great point, and you're exactly right. mRNA is continuing to expand and grow, and it's simply due to the fact that obviously you have mRNA vaccines, cell and gene therapies. There are a lot of newer technologies and newer therapies being developed that are relying on mRNA technology. Our goal is to provide the critical input proteins to create the mRNA, things like DNase-1, DNA ligases, RNA polymerases, RNase inhibitors. These are essential input proteins that you must have in order to create mRNA to exert the functions that it needs to create the other, downstreams, whether it's a monoclonal or whether it's a mRNA vaccine. These are critical input components, and they're extremely high cost, and it's a very large and growing market. So our first foray into that is going to be DNase-1. We're currently scaling that up, and we're going to look to start direct sale of that in the third quarter of this year.
Okay, well that's exciting. And there are a whole bunch of different products that you mentioned there, and I think you alluded to the fact that they have really high margins. And I think we went in a previous conversation you had said just a gram of this is worth quite a bit.
Yes.
Tell us about the value of these relative to their weight or the amount produced.
Yeah, and that's a great
more valuable than gold.
Yeah, and in some cases it absolutely is. If you look at the mRNA molecular biology reagent categories, what it's called, that's where these enzymes that build mRNA sit. It's roughly a $4 billion market, growing at 10 to 12% a year. The key is the proteins and enzymes in the system can be sold for anywhere from $400 a gram to tens of thousands of dollars a gram, depending on the grade and application it's manufactured at. So if you're using it to make an mRNA, you have to manufacture at what's called GMP. That is a much higher price point than if it's just a research grade material you're just using in the lab. So as you look at it from a weight standpoint, you could fit enough in this cup, and it would probably be worth almost
more than
it'd probably be a few $1 million.
Yeah.
So it absolutely, and the reason it has to be made at very exacting standards, very high purity, and the correct activity. So obviously there's a lot that goes into making them, but the margins are quite attractive as well.
Okay, and then I think DNase-1 is where you're going to start with that.
Yep.
And then what seems, I think transferrin was another one that you mentioned. Is that another attractive? Is that number 2 kind of after DNase-1?
It is from a direct product sales standpoint. I would actually put them as equal. DNase-1 just happens to be ready now. Where transferrin, we still have to do additional scale up and protein characterization. That actually fits in a different market, the cell culture media space, which is tangential to the mRNA, because you need cell culture media components to grow things like CHO cells or to make cell and gene therapies. You need cell culture media, and transferrin is one of the higher cost agents in that or higher cost products in that segment as well.
Okay, great. Well, thanks for the conversation about a lot of the things that Dyadic is working on.
No, appreciate it. It's definitely a fun time to be at Dyadic. We have a lot of product opportunities, and we're looking forward to bringing them to market in the near future.
All right, great. Well, we had Joe Hazelton, the President and Chief Operating Officer of Dyadic.